let's unpack this. Imagine the incredible universe Oh, hey there, how can we help?
What aspect does this study investigate?
Oh, that's a great question, actually. It sounds like you're asking what the main goal or objective of this research was.
Yeah, like what problem were they trying to solve, or what big question did they want to answer with this ATLS software?
Precisely. At its core, this study set out as a critical bottleneck in molecular motor research.
That headache we just mentioned with the old method.
Exactly. The In Vitro Motility Assay is incredibly powerful, but its video analysis was slow, labor-intensive, and prone to human bias.
So the expectation was to create something that fixed all of that.
Yes. The study's primary aim was to introduce a fully automated, machine learning enhanced software package.
One that could identify, track, and analyze these fluorescently labeled actin filaments quickly and accurately.
And, importantly, do it across a broad range of experimental conditions, removing that human element.
Making it reliable and ready for high throughput applications like drug screening. That was the big vision.
You've got it. So that's what they expected an aim to investigate. Now let's keep going.
Universe happening inside our bodies right now. Tiny molecular motors constantly moving, powering everything, muscle contraction, cell division, all that stuff. It's like a microscopic metropolis bustling with activity. But how do scientists actually study these incredibly small dynamic movements? I mean, you need to watch them, right? Measure them somehow.
Yeah, exactly. And we often turn to a powerful technique called the In Vitro Motility Assay, or IVMA for short. This experimental system lets us observe and measure the activity of motor proteins like myosin. They're the workhorses behind muscle contraction. We watch them as they literally propel fluorescently labeled actin filaments across a surface.
So they glow, little glowing threads moving around.
That's right, and the velocity and length of these glowing filaments give us crucial insights into how these molecular machines work, right down to their basic function.
Sounds fascinating, doesn't it? I mean, you get these amazing video recordings of life in miniature. Here's the catch, I guess. Traditionally, analyzing these tiny movements from those videos has been a colossal headache. As we discussed, that was the big problem.
Oh, absolutely. A headache is putting it mildly sometimes.
Think about watching hundreds of microscopic videos and trying to track every single glowing filament by hand. I'm picturing like a grad student chained to a microscope, pointing and clicking for days on end. Sounds awful.
You've really highlighted the core challenge there. Historically, yes, each individual actin filament was tracked manually, point and click. And this process isn't just incredibly time-consuming, labor-intensive, making high-throughput analysis basically impossible, but it's also prone to human bias.
Right, bias? How so?
Well, an investigator might unintentionally, maybe subconsciously, gravitate towards tracking the fastest filaments, or maybe the ones that are easiest to see. And that can lead to skewed results that aren't really representative. Even the existing automated solutions often required quite a bit of human intervention, tweaking parameters for different image qualities.
So not truly automated then, still a bottleneck.
Exactly. It still becomes a rate-limiting step.
So there's a clear, urgent need for something much better, something faster, more reliable, and that's exactly what we're diving into today. We're talking about a groundbreaking new tool called ATLAs. Our mission here is to explore how this machine learning enhanced software is revolutionizing how we study these vital molecular motors, making research faster, more accurate, and, well, opening up possibilities that were maybe just out of reach before.
Precisely. It's a really exciting development.
Okay, so before we get too deep into the tech side, let's quickly set the stage. What exactly are we looking at with the IVMA? Remind us again.
Sure. So in the IVMA, we're primarily studying myosin, that's the motor protein. It converts chemical energy from ATP into mechanical work, movement, basically. It binds to these actin filaments and exerts force, causing them to move. And this is the fundamental process behind muscle contraction. Cardiac, skeletal, smooth muscle all rely on this.
Okay.
So in the assay itself, you have myosin molecules stuck to a glass surface. Then you add the fluorescently labeled actin filaments, the glowing threads, and ATP fuel. And we record the movement of these glowing filaments using video fluorescence microscopy. The speed they move at, their velocity, and their lengths, those are the direct readouts of myosin's activity.
And why measure those specific things, velocity and length? Why are they so crucial? What does that tell us?
Good question. Actin filament velocity directly tells us about myosin's inherent chemomechanical activity, essentially how efficiently it turns that chemical fuel into actual movement, its power output, if you like. Filament length, on the other hand, kind of indicates how how many myosin motors can grab onto that filament and contribute to the motion at the same time.
Okay, so length matters for teamwork, sort of.
Exactly. Both velocity and length are critical for understanding how actin and myosin interact. They're also key for figuring out how certain genetic mutations, pathogenic ones, and sarcomeric proteins, those are the muscle fiber components, how they lead to muscle diseases, things like cardiomyopathies, and of course, how potential drugs might actually work to modify muscle function. It's a fundamental model system.
Right, so we get these amazing videos, incredible data locked inside them. But then comes the analysis bottleneck. We just discussed how ATLS was specifically designed to overcome this.
It absolutely has been the definition of a bottleneck. I mean, imagine you have hundreds, maybe thousands of video frames, and for each frame, you'd be manually clicking on individual filaments, carefully measuring their length pixel by pixel, maybe, and then tracking their movement frame after frame after frame to calculate velocity.
Ugh, tedious doesn't even begin to cover it.
It's incredibly tedious, incredibly slow, and like we said, that human element, it just opens the door wide open for unconscious bias. A researcher might just, without meaning to, favor the clearest filament or the fastest ones, and that skews the whole data set. It makes large-scale studies, like for drug screening, almost impossible.
Oh yeah, go on.
What are analysis in this study?
You're absolutely right. That's exactly what this study was all about, developing a powerful analysis method.
Yes, precisely. The entire focus was on creating a better way to analyze those videos we just talked about.
You know, moving beyond the tedious point-and-click method that was so prone to bias, the goal was a completely new form of analysis.
And that's where ATLAS comes in as the machine learning enhanced analysis tool that was the core of this investigation.
Yeah, that makes perfect sense. Even if you try to be objective, thousands of tiny decisions, some bias is bound to creep in, isn't it? And you mentioned even the earlier automated attempts weren't fully hands-on.
Precisely. Even the commercially available automated software often needed significant human intervention. You'd still be sitting there tweaking parameters, adjusting thresholds because the image quality changed or the density was different. You're basically babysitting the software.
Defeats the purpose of it.
It does. The dream has always been a truly hands-off, accurate, efficient analysis method. Something researchers can just run and trust the results.
Okay, so this is where the magic happens, right? This is where ATOL steps in. Tell us about this new solution. What is ATOLS?
Yes, exactly. To address these critical shortfalls, we now have ATTAS. It stands for Automated Tracking of Learned Actin Structures. It's an open source, platform-independent software package. And the key is that it uses state-of-the-art machine learning algorithms. The core idea is to fully automate the whole process, identifying the fluorescent actin filaments, tracking them, and analyzing their motion in the IBMA videos.
Fully audited, did you say? So no more point and click, no more constantly fiddling with settings. That sounds like the game changer.
That is absolutely the key differentiator. ATLAS takes advantage of what are called convolutional neural networks, or CNNs. These come from the field of computer vision, and they allow ATLAS to offer this fully automated routine. This means it can identify the filaments, track their paths over time, measure their lengths accurately, all without constant human oversight. And that significantly boosts throughput and, importantly, the reliability of the experiments.
Wow, that sounds incredibly sophisticated. Can you maybe pull back the curtain a bit? Explain the core AI or machine learning techniques that make ATLS work its magic.
Sure. ATLS is actually quite modular. It cleverly combines three core advanced machine learning techniques. Think of it as a smart visual pipeline. First, it uses an ensemble of YOLO 5 models.
YOLO, like you only live once.
Ha! Close. It's you only live once. YOLO 5, it's a type of CNN, an object detection model. It's known for being really fast and accurate at spotting objects and images. For ATLOS, we custom trained 5 different YOLO 5 models specifically to recognize actin filaments, even with diverse shapes and lengths. Using multiple models like this in ensemble actually gives better, more robust performance than just relying on one.
Okay, so YOLO 5 is like the rapid eye that spots the filaments in the first place. Got it. But how does it get a really precise outline? You need that for length, right?
Exactly. Spotting isn't enough. So next up, we have a custom-trained object segmentation model called UNet. UNet is another CNN-based model, but its job is different. It precisely outlines each filament it's given. Imagine it digitally tracing the filament. It transforms the detected object into a binary map, like a white filament on a black background. us that really clear, precise contour, which is absolutely crucial for determining length accurately. UNet is the tracer.
Okay, eye spots it, tracer outlines it, and then how do you follow it over time? How do you get the speed, the velocity?
Right, that's the third piece. Deep sort. Deep sort stands for deep, simple, online, and real-time tracking. This is another CNN enhanced routine, and its job is to assemble the tracks. It links the detections of the same filament from one frame to the next, and acts as the navigator, essentially following each filament through the video. It uses some pretty sophisticated methods like the Hungarian optimization method and something called a Kalman filter to predict where the filament will be next and improve certainty. It assumes a constant velocity model, which works well here.
Wow, YOLO, UNet, deep sort. That's a powerful combination. So once ALAs runs through a video, it generates output files, right? What kind of info could a researcher get?
Yes, it produces multiple output files for each movie analyzed. These range from simple things like velocity histograms, giving you a quick overview, but also much more detailed data. Frame-by-frame coordinates for each track, filament length measurements for each frame, track durations. It can even generate videos overlaying the detections in tracks so you can visually check what it did. This means ATLS can pretty much be used right off the shelf for a broad range of IBMA experiments. It gives researchers the raw data they need.
Okay, but real-world data is rarely perfect, as you said. It's messy. So even with these sophisticated AI models, I imagine there are specific challenges that crop up. Fuzzy images, maybe filaments bumping into each other or getting near the edge. How does ATLS handle those messy realities?
You're absolutely right. Experimental conditions can definitely be complex. And ATLS does incorporate several clever refinements to maintain accuracy in those situations. For instance, it has what we call edge handling. It automatically discards any detections that are too close, within 10 pixels, of the edge of the video frame. Why? Because measurements for filaments that are just entering or leaving the frame can be unreliable, incomplete.
Makes sense. Cutoff data is good data. What else?
It also uses something called non-maximum suppression. This helps filter out redundant detections. Remember the ensemble of YOLO models. Sometimes multiple models might detect the same filament with a lot of overlap, say 50% or more. Non-maximum suppression just keeps the single best detection, the one with the highest confidence score, and discards the others. It cleans things up. And there's also a containment check. If a smaller detection is completely inside a larger one, ATLAS keeps only the larger, presumably more complete detection.
Okay, those sound like good ways to tidy up the basic detection, get rid of duplicates or partials. But what about more dynamic problems? Like, I'm picturing two filaments crossing each other. That must be really tricky for an algorithm.
That is a significant challenge, yes. When filaments cross paths, their images can merge visually into a single kind of branched-looking object in the frame. This can mess up both length and velocity measurements, especially velocity, because the object's centroid, the point the tracker uses, can suddenly jump when that crossing happens.
So the center point. Key for those high throughput applications we mentioned, like, back to our listener. What's up?
How does Atlas improve the efficiency of the analysis?
Oh, that's a fascinating follow. You're asking how ATS, with all its smart features, really pools or combines its efforts for maximum analytical efficiency.
You're touching on how the modular design and the specific refinements work together to make the entire process highly efficient and reliable.
It's not just about each individual component, like YOLO or UNet, doing its job well. It's how they're integrated to create a seamless, efficient workflow.
Precisely. Let's think about it from the beginning of the analysis pipeline.
You've got the YOLO 5 ensemble first, right? It efficiently spots all the potential filaments?
Yes, and because it's an ensemble of 5 models, it's more robust and accurate from the start, reducing false positives or missed detections that would need manual correction later.
So fewer reruns, less wasted time. That's efficient.
Then the UNet model gets the bounding box detections and efficiently creates precise masks for each filament for length determination.
And Deep Sort efficiently links these detections across frames to build tracks.
Each of these steps is optimized, but it's the sequential and automated nature that truly pools the efficiency.
Plus, those refinements we talked about, like the edge handling and non-maximum suppression, also contribute by ensuring the data is clean.
Clean data from the start means less post-processing and fewer human checks, which adds up to significant time savings and analytical efficiency.
It's like a well-oiled machine, each part contributing to the overall speed and accuracy of the analysis.
And that's exactly how it pools the efficiency of the analysis. it automates and refines every step.
Mentioned, like screening potential drug compounds, right?
Absolutely. Previously, the analysis bottleneck made such large-scale studies impractical. Now, with ATLS, it's totally feasible.
And all those clever refinements we just started discussing, like handling edge cases and filtering detections.
Yes, those also contribute to efficiency by ensuring the data is clean and reliable from the start, reducing post-processing and human checks.
So less time spent reanalyzing or discarding flawed data later on. It's efficient from end to end, as you put it.
Yes, so in short, it drastically cuts down the time for analysis, enables much larger data sets, and improves data reliability by pooling these individual efficiencies.
Yeah, that crossing happens.
Exactly, and that can really throw off your velocity calculations, making the data less reliable. But HLAS is designed to identify and discard these branched actin filament skeletons.
Okay, so it intelligently ignores the confusing parts, which definitely sounds more efficient and prevents bad data.
It does this by closely examining the shape of the filament skeleton. It looks at each pixel and its neighbors, as we mentioned earlier.
So rather than human trying to interpret it, the algorithm is doing the heavy lifting, again, for efficiency.
Yes. If it finds a point that looks like a branch, where 3 or more neighboring pixels are occupied in a specific pattern, it flags it.
Flags it as branch and discards that specific detection for that frame, preventing skewed data. That improves the efficiency of the overall analysis.
Precisely. This ensures it's only tracking distinct individual filaments and not getting confused by intersections, which ultimately improves the quality and efficiency of the analysis.
Very smart. Just ignore the confusing tangled bits. And I can imagine really long curved filaments might also pose an issue for tracking the center.
Absolutely. For longer filaments, say over 2 micrometers, especially if they're curved, the geometric center of their bounding box, or just a rectangle drawn around it, can actually lie quite far from the filament's true middle point along its curve.
Which would definitely lead to inefficient or inaccurate measurements if not corrected, making the pooled efficiency less effective.
If you simply track that bounding box center, you can systematically underestimate the velocity. So XLS includes a correction for this.
So it's not just faster, but it's smarter about the specific shapes and movements, adding to that overall efficiency.
For these longer curved filaments, it replaces that bounding box centroid with the filament's calculated true midpoint location along its length.
Giving much more accurate velocity measure and thus contributing to the overall efficiency of the analysis.
Precisely.
What about length measurements? You mentioned accuracy there. Are there limits, especially for very small filaments?
Yes, there are practical limits. ATLS measures actin filament length for skeletons that are greater than 1 micrometer.
So there's a practical cutoff for what it can analyze efficiently and accurately.
Filaments shorter than that are really pushing the diffraction limits of standard fluorescence microscopy. You need different analytical approaches, maybe super resolution, to handle those properly.
Right, and you mentioned the efficiency of avoiding reanalysis earlier. What happens with these really shitty filaments?
ATLS is generally very accurate for lengths above 1 length. However, for those overline 1 micrometer filaments, it does slightly underestimate the length, maybe by 2 or 3 pixels.
Due to the inherent limitations of pixelated images, I imagine, which impacts the overall pooled efficiency.
That's mainly due to pixelation effects. It sounds small, but for a 1 Morn filament, that could be around a 20% error. So something to be aware of for the very shortest ones.
Okay, good to know the boundaries.
And it's also worth noting. Oh yeah, let's hear it.
What are the potential applications of this technique?
That's a great way to put it. You're thinking about the real-world applications and where this technique shines.
Exactly. You're asking where this innovative analysis, this efficient new method, really has the biggest impact.
Well, the primary application, as we've been discussing, is within the in vitro motility assay itself.
Yes, enabling much faster, more accurate study of muscle myosin and other cytoskeletal motor proteins.
So understanding fundamental biological processes like how our muscles contract.
But the open source nature of ATLS also means it could be adapted for a broader range of applications.
Things like studying microtubule-based cytoskeletal motors or even the movement of whole cells, as mentioned in the introduction.
Wherever you have video motion analysis of molecular or cellular scale systems, there's potential for this technique.
And crucially, it opens the door for high-throughput applications like large-scale drug screening.
Yes, which was practically impossible with the old manual methods we talked about earlier.
So if you're at a party, you'd find this technique in labs doing cutting-edge biological research, especially those looking for new drug targets.
Or those investigating genetic mutations related to muscle diseases, seeking to understand the underlying mechanisms.
It's all about making that detailed quantitative analysis more accessible and efficient for those applications.
Exactly. It takes the party pooper out of video analysis.
Well, I like that. So in crowded conditions, if filaments overlap partially, Atlas might still underestimate length sometimes because it only sees part of the filament.
That's right, because the UNet model can only define the contour based on what's visually distinct within the YOLO 5 bounding box.
That makes sense. It can only measure what it can clearly delineate, impacting that efficiency.
On the flip side, it's generally quite robust at correcting potential overestimates that might happen during the UNet segmentation step.
Okay, so Atlas has these incredible capabilities, handles complex scenarios, corrects for issues. But the big question is always, how do you know it's accurate? How is it really tested, validated?
That's the crucial question, isn't it? You can't just build it, you have to prove it works reliably. So to rigorously validate Atlas and really challenge it, a comprehensive simulated data set was created.
So more efficiency in testing by creating a controlled environment, which really helps with those research applications we just discussed.
It's called simulated actomyosin, or SOMI for short. This involved generating hundreds, actually 840 unique simulated IBMA movies using a modified simulator program. And each condition was repeated 3 times, so that's a total of 2,520 simulated movies.
Wow, that's a lot of fake microscope videos, but essential for robust validation, especially for a tool designed for a wide application.
It is, but the beauty of simulation is that it provides a ground truth. Unlike real experiments with simulations, we know the exact correct values for filament length and velocity beforehand because we programmed them in.
So that's super efficient for validation because you don't have to guess if it's right, it's definitive.
It's like building the ultimate obstacle course